Hello everyone, my name is Samir Sadao from Egypt. I am a PhD student at Chinese Academy of Agricultural Sciences in China. It's my pleasure to participate at annual meeting of ADSA 2021. My supervisor is Professor Deng Bampo, a well-known animal scientist at Institute of Animal Science of the Chinese Academy of Agricultural Sciences in Beijing, China. Today, I will share with you my presentation titled Supplementing Ruminally-Protected Lysine, Methionine, or Combination Improve Milk Production in Transition Dairy Cows. The key message of this experiment that inclusion of ruminally-protected amino acid increases dry matter intake and the milk production and decomposition, as well as increase income over feed cost. The outline of this is divided in 4 parts: background, the hypothesis, objective, and the last one, result and summary. During the transition period, many of physiological and nutritional and metabolic and immunological change is occurred during this critical period of the dairy cows because fetus and the mammary gland development and the honest secretion of milk production. That is mean causing the cow to be found in negative energy balance and in negative protein balance also. Transition period is lasted totally for 6 weeks, 3 weeks before calving. This period that is named close up, and 3 weeks after calving that is named fresh period or early lactation period. Negative energy balance, that is mean increasing the susceptibility of the cow to have production disease. Because the metabolic demand and the increase the requirement of the cow at this time to support the milk production and the feed intake is lower. So negative energy balance is occur. That is mean immune suppression and increasing the metabolic disorder disease like metritis, mastitis, and lipid-displaced apomism and the lower fertility. In this case, in low ring time, most of the cow has problem in this time, likely to remove and to be culling from the herd during the lactations. So as dry matter intake in this time is lower and do not cover the demand of nutrient to support the milk production, the negative energy balance and the negative protein balance is occur. That is mean increase the lipolysis from adipose tissue to support higher energy demand and increasing the gluconeogenesis to support higher demand of nutrients. Increasing stress and the lower concentration of insulin and also lower blood glucose. As the lipolysis increase, the NEVA non-esterified fatty acid is increased and consequently the ketone bodies is increased, leading to lower immune cell function and increasing the inflammations. The liver is playing a major part in this time because liver is oxidized non-esterified fatty acid from adipose tissue to produce energy, and the incompletely oxidative part is produce ketones in bloodstream. The other part is esterified, the triglyceride is esterified in the liver, and the most of part is stored in the liver as a liver fat to store it as FLDL, very low density lipoprotein. The other part is going directly to the mammary gland to secrete it in the milk. So our hypothesis supplementation of ruminal-protected amino acid would improve the amino acid balance. That is mean increasing the bioavailability of carnitine, leading to decrease the non-esterified fatty acid in the liver and decrease the incidence rate of ketosis and increasing milk productions. our objective is that supplementation of rumen-protected lysine and methionine, increasing the milk production and improve the performance of the cow, and also to examine the effect of supplementing ruminal-protected lysine and methionine in the liver functions. Here in the experimental design, all the details about the design of the experiment and the cow and the treatment. The cow is selected based in many parameters. One of these stays pregnant and previous lactation milk production and the body condition score and parity or lactation. We used 120 cows in this experiment, divided as 30 cows per each treatment, and the experiment is performed as a completely randomized like design with treatment arranged in 2x2 factorial. We have 4 treatment to control without supplementation and rumen-protected methionine, the lysine, and the combination. And here is the details of sampling for blood and the milk and feed and cholesterol also, and measuring body condition score. This is the total mixed ration of the diet fed to the cow during the experimental period, and here is the inclusion rate of methionine during the close-up, 0.16, and during the fresh period, and here for lysine, ruminally-protected lysine during close-up and during the fresh period. And here is the chemical analysis of the diet. For rumen undegraded protein and rumen degraded and the metabolizable protein balance and the lysine, this value is generated according to the actual dry matter intake of the cows. And the calculation of this using CNCBS model, we can see here that the metabolizable protein sublet and the metabolizable protein balance is positive during the close-up in all experiment, but for the ruminal protected amino acid is higher balance than control, but during the fresh period all cow in negative balance, but still also has higher metabolizable balance and the higher intake because the higher intake of the dry matter intake, and here is the methionine and the lysine sublet to the cows. For the result, we can show here in the first table the effect of the dry matter and the energy balance and the body condition score. Most of them is significantly different due to the supplementation of amino acids, except for body condition score during the close-up and during the fresh period, and also during the overall transition period. Here we divided and analyzed the data in 3 periods, for close-up period and for fresh period and for the overall transition period. And this is show us the effect of milk production and the composition and the milk composition yield. Most of all is significant, except for feed efficiency as calculated as energy produced milk divided by the dry matter intake and the nitrogen efficiency, and a strong tendency for increased income over feed cost due to the supplementation of amino acids. For the dry matter intake is increased significantly by supplementing ruminal protected amino acid, even there are interaction of treatment and time during the fresh period and the close-up period. And the higher increase recording during the close-up for rumen-protected methionine and for fresh period for the combination. And here during the fresh period, the rumen-protected lysine and the methionine is parallel and is the same value in almost cases. And the lower one for control during the close-up and during the fresh period. And for beta-hydroxybutyrate concentration is decreased during the close-up in response to the supplementation, and a strong distance for decrease during the fresh period. The lower one value observed during the calving date and the highest one recorded at 7 days in milk, and start to decrease sharply after this period to reach up to 21 days after calving to decrease in all treatment group except for control is still around 1.4, that is mean still in under subclinical ketosis, but the supplemental cow is becoming normal and less than the value of subclinical ketosis. For milk yield and energy corrected milk is improved due to the supplementation and the control, but here there are no interaction and the effect of time and the treatment is significant, and the milk production is maximized due to the supplementation of the combination of lysine and methionine together higher than supplementing rumen-protected lysine alone or or rumen-protected methionine separately. Milk protein composition, milk production composition of milk protein and milk fat and lactose and the total solid follow the direction of milk production. Milk lactose positively responds to the supplementation of amino acid, and this is derived by the increase of dry matter intake that stimulate glucose productions. And also for lactose, total solid and the total solid yield. For milk urea nitrogen, in this experiment we observed a decrease due to the supplementation, except for the control had recorded the higher value than the supplemented cows, and the lower value is recorded around 21 days after calving, and that is mean supplementation increase the utilization of amino acids and lower blood urea nitrogen and decrease the deamination. This is to explain to us how milk urea nitrogen is decreased due to the supplementation. In this chart, here is the effect of supplementation of amino acids and somatic cell count. In somatic cell count also the treatment is still significant, but even in this case there is still a low relation between amino acid supplementation and the lower decrease that observed in somatic cell count in this experiment. For energy balance during the close-up period, all in positive charge, but in fresh period, all cow is in negative charge because the lower feed intake that not completely match the increased demand of nutrient to support the higher milk production. Except for the control group at 21 days in milk is higher than the supplementation cow, and this it may be due the lower milk production and start to increase dry matter intake for these cows. For body condition score is significantly differ between the supplemental cows and control. During the close-up is stable, and the change is started to be increased after calving. So we can see all cow is sharply decreased after calving, except for this group that cow feeds a combination of rumen-protected lysine and methionine. At that time, cows in the group feed lysine alone or methionine alone start also to decrease, but the rate of decrease is lower than the control. So we can show the minimized decrease or the minimized change of body condition score due to supplementation. But here, we can choose start from day 0 to decrease and then become stable. And after 2 weeks after calving, the cow started to gain body condition score and to replenish body condition score again. So in summary for this experiment, we can say that the feeding combination of ruminal-protected lysine and methionine improve animal performance than either lysine or methionine separately in many aspects as follow: increasing the dry matter intake, and this is leading to the increase of milk production and the milk composition and the milk composition yield, and increasing the income overfeed cost and economic and profitability of the farm, and the stability and the minimize of body condition score, and minimize the decrease of body condition score during the transition period, and improve and enhance the energy balance, and the decrease of metabolic disorder disease such as ketosis, and the decrease somatic cell count and the milk urea nitrogen. I would like also to express my sincere gratitude to my Professor Dingbampo, my supervisor, and my lab mate, and the evaluation committee of annual meeting of ADSA 2021 for their kind support and assistance. This project is funded by the International Atomic Energy Agency and for Chinese Academy of Agricultural Sciences and for Beijing Dairy Industry Innovation Team. Thanks much for your attention.